This is an overview of a recent study I conducted about gene expression biomarkers for cisplatin as a treatment for breast cancer. Over 1.5 million women each year develop breast cancer and is the second leading cause of cancer deaths among women. Breast cancer is a carcinoma which is marked by a tumor in the lobulus or ductal region of the breast, and 20 to 30% of breast cancer cases become metastatic. Therefore, it's imperative to treat cancer as soon as possible in order to attempt to eliminate the tumor. Otherwise, if the cancer has progressed into further stages, controlling or palliation may only be possible. Many times, exposure to ineffective treatment can increase the extremity of the disease. In order to create a more efficient, cost-effective treatment process, many tools such as genetic biomarkers can be used. By measuring the expression of certain genes of interest in cisplatin-treated breast cancer, similar trends can be examined between cisplatin-treated blood cells where statistically significant trends may function as a biomarker. This would allow for an early and more beneficial individualized treatment process. This pattern of cellular resistance versus cellular sensitivity to cisplatin treatment can be seen in a study conducted by Stark et al. In 2017, where 200 different genes were tested and protein expression was measured in cisplatin-treated blood cells. So, 3 different genes were chosen. Sensitivity was seen as continuous downward regulation, while resistance was characterized by a downwards regulation until hour 6, then an upwards regulation. Cisplatin is a platinum-based chemotherapy which enters the nucleus and forms adducts with 2 consecutive guanines. The new adduct within the DNA signals for HMG domain proteins, which destroys the structure of DNA through injecting phenylalanine 37. Since the structure of DNA is messed up and cannot be repaired, replication can no longer occur, thus leading to apoptosis. Cisplatin is an alkylating agent which is not cell cycle specific but is seen to cause maximum damage in G1. The 3 genes of interest were ZNF238, ALX3, and APTX. ZNF238 and ALX3 are hypothesized to increase in expression because both encode for transcription factors that help with various biological processes. APTX, on the other hand, was expected to decrease because it encodes for a protein that fixes broken strands of DNA and was seen to decrease chemotherapeutic resistance by regulating sensitivity. The blue graph of ZNF238 shows a continuous downwards regulation during the 0 to 24-hour time period. This correlates with the sensitivity line of the blood cell graph found in a study by Stark. T-test analysis was used and concluded that ZNF238 was significantly downregulated based on a p-value close to 0.5. This gene could be used as a potential biomarker because of its correlation to cellular sensitivity. Even though the expression of ZNF238 is upregulated at hour 48, it's important to note that cisplatin has a half-life of around 30 minutes, so time points closer to this value are more relevant. Based on the correlation of cellular sensitivity and the potential as a biomarker, the expression of ZNF238 can be used to help create a more efficient and cost-effective treatment process, as people with downward expression of ZNF238 should use cisplatin to treat their breast cancer. APTX is a highly complex expression pattern and thus should not be used as a biomarker. The expression of APTX did not follow a trend towards sensitivity or resistance that was demonstrated by cisplatin-treated blood cells. ALX3 was not expressed in breast cancer because it was undetected by qPCR.